So I guess the first question is why? How come we're not getting exactly the amount? And part of that is that the membrane isn't perfectly there will be some hydrogen ions that sneak across, basically, without actually going through the ATP synthase. They either just leaky across the membrane, or they might be involved in transporting other things. So, for example, one of the things you have to do, we're making this ATP, and that's in the mitochondrial matrix, right? But now, most of the ATP hydrolysis reactions are taking place out in the cytosol. So that ATP actually has to get exported through the inner mitochondrial membrane. It gets cleaved into ADP plus phosphate, and then that ADP plus phosphate has to make its way back into the to the mitochondrial matrix. Those transport processes, especially the transport of phosphate, will dissipate some of that electrochemical gradient. And so, so basically, all the hydrogen ions that are moved don't necessarily make its way back through the ATP synthase. The dam leaks a little bit. I always think of the dam. Anybody like drive to Monterey and go up over the Pacheco Pass? And if you go up over the Pacheco Pass, there's the San Luis Reservoir there. It's a great big reservoir. It's kind of cool reservoir because, you know, most reservoirs like, you know, Folsom Lake and all of that, they collect water from the watershed that's above and control the flow. That one, no, actually what it does is it collects water that's been pumped down there, and it's just a storage. They pump it up when in the wintertime and fill it up in the wintertime, and then they let the water come out. And when it takes electricity to pump it up, and then when they need it for irrigation processes, they let it go back out, and they run electrical generators when they let it back out. I always think that's a little bit like electron transport, oxidative phosphorylation. Electron transport is pumping the water up above the dam, and then when it flows back through, it's driving these turbines to be able to create ATP. Well, the mitochondrial dam leaks a little bit, so it's not exactly perfect. It also explains why this process isn't entirely stoichiometric. If you go back and look at your basic biology textbooks and in biochemistry textbooks, often when they present the overall oxidation of glucose, say glucose plus O2 is CO2 and water, they'll link it with a specific number of ATP made, usually 34, 36, 37, like this. Well, and you'll see that variety because there isn't a hard and fast number. It's not a stoichiometric process. It depends on different things. Okay, so that leads us into the last thing I want to cover in all that is this concept of uncouplers. So these are molecules that let H plus return to the matrix bypassing the F1, F0 ATP synthase. They're basically molecules that allow the H plus to go back through. Now there's some natural ones. One of the ones natural ones are known as uncoupling proteins. And there are several isoforms in humans and in mammals. You think, well, why, what the heck would you want to do that, right? Because now you've gone through, you've complete oxidized glucose, made all this NADH, run the electron transport chain to create this electrochemical gradient. Now you're just going to let it go to waste, go to nothing. Okay. Well, one of the reasons, if you're a hibernating mammal, okay, and you're basically sleeping for a long time in your bear den, I guess, in the wintertime, and you in the wintertime, and you're not very active, and so you need to be able to generate heat, right? If I get cold, what happens if I need to warm up, right? You start shivering, right? You start muscle contracting, that generates heat and that helps keep you warm. But if you're sleeping, if you're a bear sleeping in the wintertime, you're not doing that. So they have a special fat called brown fat that is enriched in mitochondria, and the mitochondria express these uncoupling proteins. So they go through the whole metabolism process, and all they're doing is generating heat to keep the organism warm. Actually, there is brown fat in humans, especially as infants. You get brown fat, and I think as adults we retain a little bit, like maybe in our collarbone. Anybody take an anatomy class where they talk about brown fat at all? Brown adipose tissue? I think there's a little bit in adults, like around like this. And the fat is brown because the fat cells have mitochondria in there. Just like the dark meat have a lot of mitochondria, the fat has a lot of mitochondria in there. And it's just used to generate heat. So that's a natural one. The other example I'll use is a chemical uncoupler. So this is natural here. Oh, by the way, there are a lot of things, in terms of body mass index, that there is a fair amount of genetic control of body mass index. A lot of it is just determined by our genes. There's a lot of gene-environment interaction there as well. But one of the genes that's associated with obesity is differences in uncoupling, genes that encode uncoupling proteins. So basically, if you don't express or express a non-functional uncoupling protein, your basal metabolic rate will be less than if it's higher. Okay, but the other example I was going to give is this molecule called 2,4-dinitrophenol. And what is that? Well, let's see. Let's draw the structure here. Phenol, 2,4. There's a nitro. 1, 2, 3, 4. There's an NO2 there. Well, this has a pKa that's not far from physiological pKa, and so the base form of that looks like this, and it can pick up a hydrogen ion, like so. When it picks up that hydrogen ion, it becomes permeable to the inner mitochondrial membrane. So if here's our inner mitochondrial membrane, again, here's our IM space, intermembrane space. Here's our matrix. Here's our electron transport chain. Has spent a lot of work generating this hydrogen ion gradient. Well, this protonated form can actually go across the membrane and now it's on the other side. And it can dissociate. Sorry. And so the net result is we move a proton from one side to the other. Okay. So I bring this up because this was actually used as a diet drug in the 1920s, I want to say, in the 1920s. It was a diet drug. So you could basically, if you ingested this and get into your system, then your basal metabolic rate is going to go up. Actually, your temperature goes up, and you spend a lot of energy, just kind of wasting it away making heat, but you're not, you're not storing it away as fat. So it's basically a diet drug. The problem is what's known as the therapeutic index. The therapeutic index is the difference in concentration between what you get the desired effect and the effect that kills you. And the therapeutic index of this is pretty narrow. And so that it's easy to overdose and die, basically, by taking a little bit more. I mean, you're messing with the electron transport chain, right? And oxidative phosphorylation, right? So it's easy to take a little bit too much and die. The other thing too is, and I don't know exactly the physiology of this, is because it was outlawed after the 1920s, and there were a lot of women who were taking it, and one of the effects was blindness. It's like if you didn't get the dosage right, they became blind. The other reason I like to bring this up is because you still see it discussed in bodybuilding websites. Because the bodybuilding websites will promote it as a way to get more leaner. And it's one of these things. The last time I looked, like a place says, "Oh, this stuff is terrible, don't use it." But if you want to buy some, here's something like that kind of stuff, and that kind of thing. So, and I don't know, every year or two, I didn't search before coming down here, but a lot of times right before class, somebody dies from overdosing on that stuff. Anyway, so that's a chemical. This is an example of a chemical uncoupler. All right. Did we make it through there? Did we make it through this? I think this is the end of the stuff. You guys gonna have all this down for tomorrow morning, right? Yeah, I know this is fast. This is fast. Okay, so uncouplers, that kind of finishes up the stuff on electron transport, oxidative phosphorylation. This is the last of the stuff that'll be covered on the test tomorrow, and we will go. Don't get too, just take it sort of step by step through the study guides. You'll get there. All right, so let's take a 5-minute break or so, and then we'll come back. And we do have to keep moving, and so we're going to introduce the electron transport chain of photosynthesis, which will not be on the test of tomorrow.